Thank you everyone for coming again for this occasion, which is the 4th annual Mark W. Babyatsky, MD Memorial Lecture. Thanks to the Department of Medicine for supporting this very special occasion. And we certainly wish that all of you could be with us in the same room as we were in years past to remember Mark. Many of you who have been coming over the years are friends, colleagues, mentors, students and mentees here to honor Mark's memory, and Mark's family is here as well in attendance. So I want to spend just a few minutes remembering Mark and what he meant to us. And Mark was an extraordinary person, raised in a tough neighborhood in the Bronx, but he really rose out of those circumstances, attending Bronx High School of Science and then getting his bachelor degree from Columbia College, then his MD from Einstein as an AOA. And in the next 4 years, he came to Mount Sinai, where he was resident and chief resident, and much loved chief resident. His impressive performance as a resident was recognized by the legendary Kurt Isselbacher, who unfortunately passed away just this past year, legend in medicine and GI, as well as Daniel Podolsky, who recruited Mark to the GI fellowship program in the storied gastrointestinal unit at the Massachusetts General Hospital. After his fellowship, Mark continued at Mass General as a junior faculty member. And in 1994, Mark returned to the Division of Gastroenterology at Mount Sinai and continued his studies on mechanisms of mucosal repair and IBD. Although he was a success in the laboratory, Mark's true calling was really as a cherished and distinguished educator. He won numerous teaching awards at every level of the medical school and was known for mentoring medical students, residents, fellows, and postdoctoral scientists. And in 2002, Mark became the director of the internal medicine residency program and co-program director of the internal medicine pediatrics residency, as well as vice chair for the Department of Medicine for education. And in the next 8 years, Babs, as he was fondly known, nurtured and promoted the careers of over 400 residents and fellows. His teaching rounds, affectionately known as Babyatsky rounds uniquely, were the highlight of the week for all interns and residents who knew they would walk away with many clinical pearls, new insights into all types of diseases. Mark also earned national recognition among internal medicine program directors and became founder and director of the Education Research Consortium of Program Directors and worked with the ACP as a member of the in-training exam and education research committees and as an ACP representative to the NIH NHGRI Genomics Consensus Conference on developing a blueprint for primary care physician education in genomic medicine. In 2010, Mark became chair of the Samuel Bronfman Department of Medicine and professor of medicine at Mount Sinai. And in this capacity, he made an impact on the educational and research missions of the department, as well as on patient safety and quality improvement programs, community and multicultural affairs, and faculty practice operations. In early 2014, he assumed new challenges as chair of medicine at Monmouth Medical Center and St. Barnabas Medical Group, South Region, to develop systems for providing high quality and cost-effective care. And Mark, as a person, was remarkable in so many ways. He had a photographic memory, an encyclopedic knowledge of not only medicine, but also arts, theater, science, and politics. And if you asked him about a movie, he had not only seen it, he could recount everything about the director, producer, actors, already read the book, knew if it had won Academy Awards, and so on. He also embodied the keenest doctoring skills, an innate ability to mentor and guide trainees, really an effortless talent for teaching, and an incredibly accessible body of knowledge for medicine and life. He truly cared about everyone that he touched and always asked how you were as his first question to you when he greeted you. And this was really a heartfelt expression of his concern. He exuded optimism, warm humor, friendliness to all people of all walks of life. He had a profound impact on myself at critical junctures of my life, and I know that I am not alone in that. And surely we are all better for having had Mark among us, and we miss him deeply. So now, in his memory, it's my distinct pleasure to introduce the 4th annual Mark Babyatsky Lecture, Dr. Timothy Wang. Dr. Wang is chief of the Division of Digestive and Liver Diseases and the Dorothy L. Daniel H. Silberberg Professor of Medicine at Columbia University Medical Center. Dr. Wang received his undergraduate degree from Williams College summa cum laude and his MD from Columbia, followed by medicine residency at Barnes, WashU, and then GI fellowship training at Massachusetts General Hospital, Harvard Medical School. He joined the faculty there and quickly rose to the rank of Associate Professor of Medicine before moving to University of Massachusetts Medical School, where he was chief of the Division of Gastroenterology and the Gladys Smith Martin Professor of Medicine. And in 2004, he moved to Columbia for his present role. In addition to his role as chief of the division, Dr. Wang is director of the Tumor Microenvironment Program in the Irving Cancer Center and director of research for the Pancreas Center. He has received numerous awards for his groundbreaking research in GI cancers and GI gastric physiology in particular. He's won the AGA's Robert and Sally Funderburg Gastric Cancer Award and the highest honor of the AGA, the William Beaumont Prize in Gastroenterology. In 2016 and 17, he served as president of the AGA, and he has also been an associate editor of Gastroenterology. Tim has been funded continuously throughout his career for research in GI cancer and physiology with numerous R01s, UO1s, and a merit award. He's authored more than 280 original manuscripts and has left his mark as a mentor to dozens of trainees, which is something that Mark Babyatsky certainly would also have cherished. Dr. Wang exemplifies the very best of academic gastroenterology and was also a dear friend of Mark Babyatsky. And Tim, I want to thank you for joining us today to honor the memory of our friend Mark Babyatsky. Thanks so much.
Thank you, Bruce, for that very kind introduction. And it is a great pleasure to present at this medical grand rounds in honor of my friend, Dr. Mark Babyatsky. Mark was an outstanding physician and educator, and as outlined by Bruce, an inspirational leader who made many contributions to the field. And it's an honor to give this talk in his memory. Mark and I were fellows and then junior faculty together at MGH. And the topic I chose today, acid suppression and gastric disease, stem cells, peptic ulcers, and cancer, is given in part, or inspired in part by Mark. So I do have some disclosures here to note. I've been a consultant for Novo Nordisk, GlaxoSmithKline, and most notably recently AstraZeneca. So I will be discussing proton pump inhibitors today, and thus my relationship with AstraZeneca is relevant, and my comments need to be considered in this light. So to start off with, I'll go back to 1988. This is actually 1989, a few months before Bruce arrived on campus in Boston. And so I met Mark Babyatsky in 1988 when he joined our GI fellowship at MGH. And this is him shown here in the blue. That year, I was a third-year GI fellow, and Mark was fresh from his chief residency at Mount Sinai. And he came to Boston really full of energy and enthusiasm, and as Bruce noted, a very outgoing, extroverted individual who had great stories about his time in New York and about growing up in the Bronx. And he chatted up very well many of the fellows, many of the medical residents. And quickly he became the major cheerleader for our division, convincing record numbers to go into GI. He quickly became our best recruiter. I think he convinced Bruce to come to MGH for fellowship. And that year actually was the last year that Kurt Isselbacher, shown here in the front, was chief of the division. And he quickly transferred that next year, the chief job to Dan Podolsky, also shown here. And Dan, I believe, was the Babyatsky lecturer last year. Also shown here, sort of in the middle, I don't know if you can see my pointer, but is Anil Rusagi, who gave the Babyatsky lecture, I think, in 2018. Anil is currently the Cancer Center Director and also acting Dean at Columbia. So at MGH, after his clinical year, Mark joined the laboratory of Dr. Steve Brand, shown here in the front on the right. And he worked in the Gastrin laboratory, as outlined here in the title of the slide. So we overlapped for several years working on Gastrin biology and other projects. Steve Brand has since retired, but also shown here are Juanita Merchant in the front, who is now a division chief at the University of Arizona, along with Loyal Tilton in the back, and Dan Chung, who remains at MGH and was for many years the clinical chief there. So in 1994, Mark returned to New York City and to Mount Sinai. And I would just like to summarize a little bit of some of the work that that he did and we did together. Mark was very productive actually as an investigator in the 4 or 5 years that he was at MGH, with more than half a dozen publications, some of which are shown here. I was fortunate enough to be able to collaborate with Mark on several studies related to Gastrin, but he also worked on trefoil proteins. These are PEP proteins secreted by mucous cells that provide gastric and intestinal cytoprotection. His best known study, which I'll discuss later, was this work that he actually did with Dan Podolsky, whom he collaborated with, and on various types of ulcer models. So in 1994, Mark returned to New York, but he remained with a strong interest, as Bruce outlined, in ulcerative lesions of the GI tract. He gradually worked a little bit more on IBD, but early on, much of his work was on gastric biology, trefoils, and ulcer healing. So to honor Mark, then I thought I would talk a little bit about one of my interests. As Bruce mentioned, my major interest in my laboratory is on GI cancers of all types, but I've worked a little bit on peptic ulcer disease, and particularly recently, thinking of Mark. And so I thought I'd start off this lecture by reviewing just some of the basic aspects of ulcer healing, and then bring in some of the latest research and findings from my laboratory and others. Now, peptic ulcer disease, as many or most of you know, has many causes, but the primary etiologies are Helicobacter pylori infection and non-steroidal anti-inflammatory drugs. Back in the 1980s, when Mark and Bruce and I were in training, the topic of peptic ulcer disease was a very actually active area of investigation. H. pylori was discovered in the 80s. PPIs were introduced in around 1989. Now, we would like to distinguish between erosions, as shown here on the left, which are smaller, shallow breaks in the mucosa that reach but do not penetrate the muscularis mucosa, and ulcers, which are full thickness breaks that do penetrate the muscularis into the submucosa and therefore can reach some of the larger blood vessels and cause GI bleeding. Now, the ability of the gastric mucosa to regenerate after such an injury, such as an erosion, really depends on whether or not these lesions go on to form full-blown ulcers. And this concept of regeneration and wound repair is now an active area of investigation in numerous organ systems and a bit in the upper GI field. So in animal models, we and others have studied several models of stomach ulcers or gastric ulcers using NSAIDs, alcohol, acetic acid, and cryo-injury. And we'll be discussing a few of these models. Shown here on the right is the acetic acid ulcer model that I'll be discussing a little bit later. So based on these experimental model systems, we now have defined at least 4 different stages of ulcer healing. And some of these are also of interest to the IBD field, in particular Dan Podolsky's laboratory. So first is this stage of restitution. Gastric epithelial cells that are bordering the ulcer migrate across to restore the damaged barrier. And the process is independent of cell division, requires blood flow and alkaline pH in the stomach. And there are ways to model this in the laboratory. In particular, the Podolsky lab had something called the razor blade assay, which Bruce probably remembers. Next, there's the recruitment of inflammatory cells, this influx of white blood cells, the expansion of stromal niche cells, an increase in angiogenesis. Third is this stage of proliferation, where there's increased cell division by active progenitors, producing progeny or daughter cells that are able to then go on and fill these epithelial defects. And finally, there's the stage of stem cell activation, which I'll be speaking on a little bit, enhanced activity by normally slowly dividing gastric stem cells. So what inspired me recently to think again about peptic ulcer disease was this earlier work by Mark Babyatsky, who was really one of the first at MGH to look at the healing of stomach ulcers. And while at MGH, he collaborated, as I mentioned, with the Podolsky lab, as shown here, which was primarily interested in IBD and intestinal ulceration. So it was actually Mark who suggested the study of examining the role of trefoil peptides in the healing of stomach ulcers. These trefoil peptides are these 3-looped cloverleaf-like peptides. And one of them was discovered by Dan Podolsky, called ITF at the time, now TFF3. And the major one in the stomach is this other one called HSP, for human spasmolytic polypeptide, or now called TFF2. So Mark asked the question about what role these might have in the healing of ulcers in the upper GI tract. And so he utilized rat models of stomach ulcers, as shown here, ethanol and indomethacin-induced gastric injury. And he used these two tripeptides, ITF and HSP, and he gave them by oral gavage and IP injections, and found that these tripeptides produced by mucous cells in the gut protected against ulcers by effects on restitution and proliferation. So in thinking about Mark in the last several years, I became interested once again in stomach ulcers and in the role of healing of these, particularly by stem cells, which has long been an interest in my laboratory. So just a very brief background on gastric stem cells. The stomach can be divided anatomically into two sort of major organs in a way. The larger gastric corpus, which accommodates food and liquid and secretes acid, and the gastric antrum, which is the muscular region of the stomach, which grinds foods and empties then into the small intestine. They're both filled with gastric glands, which are shown here in these little cartoons, which are sort of little test tubes that are narrowed in the middle in this region called the isthmus, where the stem cells and cell division occurs. Now, stem cells in the isthmus undergo bidirectional migration to go up to the gastric pits and down to the base of the gastric glands. And we and others have identified a number of markers for both antral and corpus stem cells. And I'll be discussing briefly the corpus stem cells, in particular, a marker, a transcription factor called Mistl, which marks these corpus stem cells, and the antral stem cells, which are marked by a gastrin receptor, the so-called CCK2 receptor. And as you'll hear also, the CCK2 receptor is also expressed by many cell types in the corpus, in particular progenitor cells and ECL cells. And I'll be discussing that a little bit later in my talk. But the questions we wanted to ask about gastric stem cells and progenitor cells were, what role do stem cells play in ulcer healing? And what is the effect of PPIs and Gastrin on some of these stem and progenitor cells? So I'll just start off by talking a little bit then about the corpus stem cells that I mentioned, which were marked by a gene or a transcription factor called Mistl, which is a basic helix loop helix transcription factor, previously known to be, best known to be expressed in Schüff cells, which are secretory cells at the bottom of the corpus glands. But we found that Mistl also marks the corpus stem cell. And this stem cell had previously been identified by several investigators, Karam and LeBlanc, as morphologically as a small, poorly differentiated, granule-free cell. So we used this genetic model where Mistl was tagged to this Cree-ERT, which allowed these cells to be marked by red or green markers. And so we could follow these cells and their daughter cells over time. And we found that indeed it marked a single solitary cell in the isthmus of the corpus, as shown here in immunostaining or by green fluorescence, and that these cells divided very slowly, perhaps every 5 to 7 days, but eventually lineage traced the entire corpus gland. In addition, in working on models of diffuse gastric cancer that I won't get into, we found that these corpus stem cells are supported by a number of niche cells, supporting cells that secreted factors that made survival and stemness of these cells in the corpus possible. And these included endothelial cells that express a chemokine called SCF1 or CXL12, and innate lymphoid cells, which have been also much of interest to the IBD field. These were ILC2s that expressed a Wnt factor, an atypical Wnt called Wnt5a, and these niche cells appeared to regulate the corpus stem cell. So having identified these corpus stem cells and becoming interested again in sort of the mechanisms of repair of peptic ulcer disease, we set out to use our Mistl Cree-ERT transgenic mice in several models of gastric ulceration, which are well suited particularly for mice. And these included two models. One is the acetic acid model, and the other is the model of Helicobacter infection. Now, in the acetic acid model, as shown here in this little cartoon, and this was created by a group in Japan, acetic acid is applied for 60 seconds, and we just did this corpus model, and it's to the serosal surface of the corpus using this tube, the little straw-like device containing a cotton ball that's dipped in 100% acetic acid and applied for about 60 seconds, inducing this ulceration. Now, the ulcer typically regenerates in about 30 days. Now, in the other model, we infected mice with Helicobacter felis, which leads over several months to chronic and active inflammation of the mucosa. So these two models were then used to study the contribution of Mist-1 stem cells to epithelial regeneration. It gives these ulcers, sort of as you can see here with acetic acid. So we found that in response to acetic acid, and this is the first model of acetic acid-induced ulceration, the Mist-1 stem cell lineage traced significantly in response to this injury. On the left, at the top, you can see the entire stomach splayed out, and the ulcer was given here in the corpus. And you can see adjacent to that ulcerated area, there's marked lineage tracing, which is typically not seen in this model for 6 months or more. So in this cartoon here, you can see on the left the regenerated epithelium, on the right is control. And again, you can see in the control, there's sort of partial tracing with some cells expanding around the isthmus, which is typical of these slowly dividing stem cells. But in the acetic acid injury, you see marked tracing, almost entire glands, and you can sort of quantitate this and find that, in fact, there's significant 3- or 4-fold increases in activity of these quiescent stem cells in response to the injury. Now, we also looked at Ki67 staining and found, as shown here on the right, a marked increase in proliferation, typically in this isthmus area that correlated to a large extent with lineage tracing by these Mist-1 stem cells. Notably, we found that at the wound edges, when the tracing occurred, these basal chief cells with tracing at the bottom tended to be lost. So that's the acetic acid model. Now, the other model was the Helicobacter infection model, and we saw very similar things, which is increased lineage tracing from these Mist-1 quiescent sort of corpus stem cells. So again, in response to infection, marked lineage tracing, partial or fully traced glands that are seen in the Helicobacter infected mice at 90 days, with only a few partially traced glands in the controls. Similarly, here we show staining for the chief cells with immunofluorescence for the intrinsic factor, which is very prominent in the base of control glands, but again tends to be lost. And also, as not shown here, Helicobacter infection led to an increase in proliferation, particularly by the trace cells from the Mist-1 stem cells. So why does this activation of corpus stem cells occur? We began to look at some of the niche factors that we know that are released and that regulate these corpus stem cells, and this includes Wnt factors. And Wnt factors are able to activate typically beta-catenin, but Wnt is an atypical Wnt and has been linked in many ways to the activity of stem cells. So we looked at all the different Wnt's that were expressed in the corpus of the stomach, and Wnt5a, as shown here, was the highest expressed in the regenerating gastric epithelium. And it was elevated about 2-fold in ulcer disease compared to the control stomach. Now, we asked the question, how important is this particular Wnt, Wnt5a, this atypical Wnt, in ulcer healing? So we carried out a conditional knockout using this particularly CRE-ERT Wnt5a flox mouse that I won't get into. So bottom line, we induced knockout of Wnt5a, and this led to significantly impaired ulcer healing, as shown here, with much larger ulcers seen compared to the control mice. And we actually quantified the area in these knockout mice, showing 3- or 4-fold increase in ulcer area compared to the control mice. Now, Wnt5a, we confirmed, was located in the area of these corpus stem cells, as shown here by in situ, and we also confirmed that the Wnt5a receptor, Frizzled5, was highly expressed in the corpus stem cell. So this is one pathway, certainly, for activation of these quiescent stem cells. Now, where does the Wnt5a actually come from? Well, we actually, in a previous study, had localized much of the Wnt5a in the stomach to these innate lymphoid cells, so-called ILC type 2s, which are express the surface receptor CD90.2. And we again confirmed in our model that the CD90.2 positive cells were localized in the corpus, in the stem cell zone, very close to the Mist-1 stem cells, as shown here with the ILC2s in green and the Mist-1 stem cells here in red. And when we looked again at the regenerating epithelium, we saw more red lineage tracing, of course, but we also saw an increase in the abundance of these green innate lymphoid cells in the area of the proliferative zone of the isthmus. So in response to ulceration, there's a recruitment not only of macrophages and neutrophils and other immune cells, but in particular, ILC2 cells, which appear to expand. So when we depleted these innate lymphoid cells using an antibody to CD90.2, and therefore depleting these ILC2s not only from the blood, but also from the stomach, we were able to see impaired ulcer healing in our acetic acid model. So to summarize this part of the talk, which is focused on peptic ulcer disease, MIST-1 labels isthmus stem cells in the gastric corpus, which under homeostasis are generally quiescent, rarely dividing, but in response to significant injury, such as peptic ulcer disease or simply Helicobacter infection, these quiescent stem cells become activated and they contribute to the regeneration and the response to injury. Now, these stem cells appear to be regulated by Wnt5a, which controls their proliferation. And with depletion of Wnt5a, there's reduced ulcer healing and tracing. Wnt5a is mainly made in the gastric corpus by innate lymphoid cells, which are niche cells for the corpus stem cell, and express the receptor Frizzled5 highly in this particular stem cell lineage. And so we can think going forward in the future about therapies perhaps aimed at activating these stem cells through manipulation of Wnt5a or the receptor Frizzled5, or even through the recruitment of innate lymphoid cells as another strategy for aiding ulcer healing. So I'll now switch gears a little bit and talk a little bit about acid suppression and get into a little bit of a discussion of proton pump inhibitors. I thought I'd take us back through the early part of the 20th century with the initial observation that, yes, peptic ulcer disease is almost by definition induced by acid, and it was Schwartz back in 1904 who said no acid, no ulcer. We can think about all the various therapies that were used for peptic ulcer disease in the early part of the 20th century. But in the mid part of the 20th century, really the only drugs available were treatment with antacids, and peptic ulcer disease was very much a surgical disease with various forms of ulcer surgery, particularly Bell-Roth 2 and others, in order to manage patients with severe and refractory peptic ulcer disease. So the first movement forward in terms of therapeutics really was in 1972 when the drug cimetidine was developed by Sir James Black, and these were the first targeted drugs for acid inhibition. The next 20 years or so really was the growth of H2 receptor antagonists and H2 blockers. And then the 1980s came about, and our thinking about peptic ulcer disease changed with the discovery of H. pylori infection, the early introduction of eradication regimens, but at the same time, there was the development of testing of omeprazole, the first proton pump inhibitor. And this development of this drug was slowed and almost stopped because of the finding when testing in rats, toxicity testing, that these animals developed ECL hyperplasia that was severe, and in fact, they developed ECL cell carcinoid tumors. And there was a lot of great concern at the time, but looking at several other models, including the human model, was the finding that there was some ECL cell hyperplasia, but no carcinoids seemed to develop. And so the drug was approved by the FDA. Several other drugs fell by the wayside because of this concern. But this was a bold move at the time by Astra, and in fact proved to be a good move. The drug was approved, and this drug really caught on in the early and mid-1990s. So we saw over the last 20 years a decrease in ulcer surgery, the rapid growth of PPI use, not only for peptic ulcer disease, but for GERD, and then as I'll talk about non-ulcer dyspepsia and ulcer prevention. So just a little bit about how these drugs actually worked. And some of this work was really carried out in the 1980s by Dr. George Sachs from UCLA, who sadly passed away in the past year. But we found out that these PPIs work by binding irreversibly to the alpha chain of the hydrogen potassium ATPase pump, but it binds to this pump only when these pumps are actively secreting. So just a little bit of cartoon cell biology. These pumps are normally present in these tubular vesicles in the resting state, little vesicles in parietal cells. And in response to stimulation, primarily cyclic AMP stimulation secondary to H2 receptor signaling, these tubular vesicles then fuse with this resting secretory membrane, and then have active secretion of hydrogen and access to the lumen of the stomach. And this occurs again in response to eating. So of course we say that these drugs should be taken prior to eating to 30 to 60 minutes, so that when they're in the serum and when they're in the circulating throughout the body, they have access to actively secreting pumps. So these the drug has a short half-life, but a long biological half-life, because it irreversibly inactivates the hydrogen potassium ATPase. And it takes about 3 to 4 days after stopping the drug, before acid secretion restored. So PPIs are prodrugs. They're weak bases that at a neutral or alkaline pH are non-protonated and readily cross cell membranes, including into parietal cells. Now, in the highly acidic environment of the parietal cell, they pick up a hydrogen, they're protonated, and they're trapped within parietal cells. And so this drug builds up 1,000-fold or more inside of parietal cells. And in response to protonation, they undergo this sort of morphologic chemical change, where they become this active moiety, a thiophilic sulfonamide, which undergoes this sort of rearrangement, and then eventually binds to the hydrogen potassium ATPase pump through the cysteine residue. So it forms this stable covalent bond, inactivates the PPI, and is able to block acid secretion. So in the last 30 years, we've seen the introduction not only of omeprazole, but also several other PPIs. There are now 6 different proton pump inhibitors available in the US. In addition to omeprazole, there's lansoprazole, pantoprazole, rabeprazole, along with esomeprazole and dexlansoprazole. So these last 2 are enantiomers, with esomeprazole being the S enantiomer of omeprazole and dexlansoprazole being the R enantiomer of lansoprazole. There are also oral and IV formulations. There are immediate release PPIs that are combined with sodium bicarbonate. And I'll just mention very briefly, there's a new proton pump inhibitor, vonoprazan, which has been approved in Japan. It's a different molecule. It's a first-in-class competitive potassium acid blocker. Not yet FDA approved in the United States, but again, approved in Japan and apparently very effective. So following their introduction, the PPIs steadily increased to the point they became the 3rd largest class of medications prescribed in the United States by around 2010. And around that time, there were more than 100 million prescriptions filled annually, with about 5% of the US population using on a regular basis PPIs. So that's a lot of prescriptions. And in part, this was because PPIs were felt to be extremely safe, and the indications started to broaden, as they do for many pharmaceuticals, so that patients not only with peptic ulcer disease or severe GERD or other reasonable indications were getting the drug, but patients with mild GERD and non-ulcer dyspepsia, for which the efficacy is somewhat questionable, gastric cytoprotection, for patients at increased risk for peptic ulcer disease, which became a fairly broad category. And for many patients, as we found out, were prescribed PPIs in the hospital, and then the drug was never stopped. So this was actually, PPIs became a marker of patients who had been recently hospitalized. And a lot of this work recently has been done by Dan Friedberg in our division. So this became an issue, that these drugs were to some extent, I would say, overprescribed. So thus, with so many patients taking PPIs, and with the development of newer sort of electronic medical records and electronic prescription records, this allowed tracking of such patients who were on PPIs. They became sort of their own sort of class of patients that could be followed. And this became a goldmine, really, for case control or cohort studies. And this is not to blame clinical investigators who do these sorts of epidemiologic studies, as they became very easy to do, but the literature grew rapidly, with now thousands of papers published on associations between PPI use and possible adverse effects. So this is, I found one recent publication, so not so different from many of the others. And the list here of sort of associations is actually longer than this, but this is a reasonable sort of quick summary. And again, the things to keep in mind are that this data is largely observational. They derive almost all from case control studies that are able to indicate this sort of association. I would say that, and here in this particular study, they grade them as whether they're reasonable or out to lunch. But the one association that seems to be real certainly is this slight increase in enteric infections, particularly Clostridium difficile infection, which I think is real and but is still less than seen with antibiotics. And the reason for these sorts of infections is that there's reasonable evidence that PPIs, by inducing hypochlorhydria, do result in mild changes in the GI microbiota, in particular changes or loss in potentially some protective bacteria that might prevent such infections. Most worrisome to the lay community has been some of these associations, including dementia, Alzheimer's disease, severe cardiovascular disease, that means myocardial infarctions, strokes, and of course, cancer of various sorts. But the few randomized, most of these are unlikely to be real. They're probably attributable to bias rather than causation. The few randomized controlled studies that have been done have not found most of these adverse effects. And so at least at Columbia, most of our clinical investigators have concluded that PPIs are actually a marker of greater illness or disease. But I should mention that there is this one that's probably true, which is changes to the stomach can be seen. As I've mentioned, some increase in ECL cells, some hyperplasia, and also these gastric fundic polyps probably do occur in association with PPIs. And so I'll talk a little bit about the effects then of the sort of gastrin on the stomach. So with blockade of acid secretion and loss of acid in the stomach, what happens then is you lose this negative feedback inhibition of gastrin or G cells, leading to greater gastrin secretion and hypergastrinemia. And so this then drives increased expansion of ECL cells and parietal cells to some extent. As I've mentioned, certainly in all sorts of animal models, you see the slight increase in proliferation in the proliferative zone of the corpus. And as I mentioned, rats can develop ECL cell tumors, although this does not really occur in patients. But you do see this sort of hyper expansion of ECL cells and parietal cells, and this to some extent can make it then harder to stop PPIs. That is, when you stop the PPIs, you're left with this increased acid secretory capacity. So as soon as gastrin is turned back on, as soon as the acid secretion is turned back on, you actually have greater acid secretion after stopping PPIs, which again makes it more difficult to stop the drug. But the question that we had was how do PPIs really cause these ECL cells to expand? Does it actually cause them to proliferate, or are there other mechanisms going on? So we went on to do this study that we published recently, which is to look at what is the mechanism by which hypergastrinemia expands gastric ECL cells. And we found, as sort of the punchline for the study, that occurs through effects on CCK2R expressing progenitor cells via ERK activation rather than actually acting directly on ECL cells themselves. And I should give credit to the study for Wei Shang and Armando Malagola, who did most of the work in this study that was published in CMGH. So the reason we were able to sort of investigate this model is because we had been working to some extent on ECL cells, a little bit indirectly, by our work on this enzyme histidine decarboxylase, or HTC. And so one of our early studies back at MGH, or when I was working on gastrin biology in more detail at that time, was we found that this enzyme was upregulated by gastrin, and it's expressed primarily in ECL cells, as ECL cells are the primary cell within the stomach that make histamine, the final common mediator of acid secretion. And so we made these HTC GFP mice, where the HTC gene was tagged with the green fluorescent protein, and we were able to see very easily these ECL cells within the stomach by fluorescent microscopy. And so we did a little experiment of giving these mice omeprazole every day for 3 weeks, and lo and behold, these ECL cells expanded. You see more green cells in the stomachs of mice where their proton pump is inhibited. And we could actually quantify this in the number of ECL cells per 60 glands. You could see over 3 to 4 weeks, it goes up pretty significantly in the stomachs of these mice. So then we were asking the question was, is this due to proliferation of these cells, or where do these ECL cells come from? So we actually combined this with proliferation markers to try to see KS67 here. I think we also did BRD to see where the proliferation was coming that was generating these new cells. And we also then looked at the expansion of these ECL cells in different portions of the stomach, whether they're deriving from these basal ECL cells or whether the green cells were appearing in other parts of the stomach. And what we found was that most of the proliferation was not in the base of the glands, it was in the isthmus region and perhaps in the neck of the gastric glands. And that the ECL cells were appearing not at the bottom and moving up, but they were appearing in the mid portion of the stomach in the proliferative zone and then appeared to be moving down. So our findings go against this idea that these endocrine cells were showing plasticity and giving rise to new endocrine cells themselves, but instead these endocrine cells were coming from a different source. So then we went back and looked at the cells that were responding to gastrin. And so we had also done some work with the gastrin receptor. This is called cholecystokinin 2 receptor. We used to call it the CCKB receptor. And it was well known to be expressed in ECL cells and in parietal cells where it mediates acid secretion. But when we looked first by in situ hybridization, we also found a significant level of expression in the mid portion of the gastric glands and the so-called gastric isthmus. As shown here, when we try to co-localize it with ECL cell markers, we certainly show some co-localization with ECL cells. But again, we would see these cells, and this is actually with a transgenic mouse where we tagged again here tomato red or red fluorescent protein to the CCK receptor. And so if you look at co-localization of chromogranin A, which is an ECL cell marker, these are all ECL cells where the red marker is expressed. So the gastrin receptors are certainly expressed in ECL cells. But again, as shown in red, there is also expression higher up in the gastric glands. And so then we asked the question about what happens when you give proton pump inhibitors and induce hypergastrinemia. And was there expansion of these red cells from the bottom, or did the red cells expand elsewhere? And what we saw, obviously, was that there was an expansion of cells in the proliferative zone of the isthmus of the stomach. And these progenitor cells that we identified would expand from the isthmus of the stomach and then expand down. And you saw lineage tracing, which we call partial lineage tracing, because this is not like the Mistl tracing where the glands were totally lineage traced, suggesting a stem cell was expanding, but in fact, a patchy expansion, suggesting that there's a progenitor, early progenitor, giving rise to other cells, which included, of course, the ECL cell. So to look at this set of progenitor function for these gastrin-expressing cells, we isolated cells from the stomachs of mice, and we isolated gastrin receptor-expressing cells in two types. One, we isolated ECL cells, so we isolated cells that expressed the gastrin receptor and HDC, and these were ECL cells, and we tried to grow them in culture. And we isolated gastrin receptor expressing cells that did not express HDC, and these were what we call the progenitors. And we tried to grow them in culture, the sort of organoid assay that's now very commonly used in Matrigel, 3D Matrigel. We found that the HDC negative cells, the progenitor cells, could easily form organoids, but the ECL cells themselves could not. And this is shown here on the right are the ECL cells, on the left are the progenitor cells that seem to grow very nicely. And then we stimulate them with gastrin. We're able to show that we could upregulate in these cells, and this is a Western blot, and you can see the gastrin stimulation here. We could upregulate both chromogranin expression. So these cells are actually then forming sort of ECL-like cells, as shown in the organoids. You can see this in fluorescence here with the chromogranin expressed in these organoids with ECL cell-like cells being formed, probably not fully differentiated, but early ECL cell progenitors. And we also upregulated MAP kinase. And so what we found doing these organoid assays is if you blocked MAP kinase, you block differentiation and expression of chromogranin. And so that suggests that in response to hypergastrinemia, you can block the CCL hyperplasia possibly by using a MAP kinase or ERK inhibitor. Here it was UO126. So going to the next slide then, this is a summary then of what we've learned about hypergastrinemia secondary to proton pump inhibitors and their effect on ECL cells. And the bottom line is that in response to PPIs, gastrin goes up and it stimulates many things, parietal cells, ECL cells, but it also stimulates these gastrin receptor expressing progenitors and activating MAP kinase pathways, and that promotes the differentiation towards this ECL cell lineage and leads to hyperplasia of ECL cells, which so far in humans with this simple PPI exposure certainly doesn't lead to ECL cell carcinoids, but potentially this observation does have some relevance in terms of the treatment of patients who do develop ECL carcinoids for other reasons, whether it's in some patient with MEN1, for example, or etc. This could have clinical relevance to patients with gastric carcinoids. So just in the last part of my talk, I'm going to go quickly through some discussion of proton pump inhibitors and some of their possible side effects, and in particular address the issue of cancer, in particular gastric cancer. And what we've seen is that PPIs can induce hypergastrinemia. They can cause some increased proliferation in the gastric corpus. And very early on, therefore, people began thinking back in the late 1980s about whether there was some theoretical concern about a risk of gastric cancer with the introduction of PPIs. And as we talked about, the ECL cell carcinoid findings sort of faded away fairly quickly. In the early 90s, these drugs were widely used, no signal appeared. But then this paper by Ernst Kuipers, published in the New England Journal in 1996, suggested that patients with H. pylori infection who were treated with PPIs showed increased progression of atrophic gastritis, which is thought to be a pre-neoplastic precursor of gastric cancer. And this led to a lot of concern and discussion back at that time. And it also led to 4 additional randomized controlled trials, which were all summarized or analyzed in Gut in 2006. And so bottom line is that these findings of increased atrophic gastritis were not confirmed. And again, the concern went away, although there's always remained some thought about the fact that PPIs do cause this shift in H. pylori infection. And so this is something we tell our fellows, which is that if you're doing an endoscopy on a patient who's on a PPI and you're looking for H. pylori, biopsy the corpus as well as the antrum, because there's actually a slight shift from the antrum to the corpus with H. pylori colonization. And so there is a thought that you could increase pan-gastric colonization with H. pylori. And there is this increase in corpus proliferation, which is part of the mechanism certainly for funded gland polyps. But there's also some recent concern about a change in the gastric microbiome. And there's now been a number of studies that have looked at effects of the PPIs on gastric on the gastric as well as the intestinal microbiome. Some studies by Dan Friedberg and our group have looked at this as well. But overall, the studies suggest that there are effects, but they're generally small, and the significance is still sort of unknown. But then this paper came out a couple of years ago, which alarmed a number of people. It was published in Gut, which is a very good journal. And they concluded that long-term PPI use increased the risk of gastric cancer in patients on H. pylori, who have H. pylori infection. And so this was a study done in Hong Kong and included patients who had received a PPI triple regimen in the 2003 to 2012. And then they followed up patients who actually failed this regimen and therefore still had H. pylori infection. I'm not sure why these patients couldn't be eventually eradicated, but they were followed up for an average of 7 years or so. And what they found was that patients who were on, so after failing the regimen, they were either on a PPI or often an H2 blocker. And so what they found was that patients who remained on PPIs had an increased risk hazard ratio of 2.44, while the patients who remained on the H2 blocker had a lower hazard risk of 0.72. And so they concluded that long-term use of PPIs was associated with an increased risk of gastric cancer even after H. pylori eradication. This had been looked at by a number of investigators, and most of the major medical societies, though, have not accepted these conclusions. This includes the AGA and the Canadian Association of Gastroenterology. This was reviewed in Gastro by Dan Friedberg and others and discussed the findings. And also the Canadian Association of Gastroenterology basically concluded that the findings were unjustified. So the main flaw seen by many of us is that all these are, again, studies that are not randomized, and that there still is major bias that results in these major baseline differences in PPI users and non-users. So important points to consider and factors that could be pro-cancer or carcinogenic are the time frame, because we know cancer takes decades to develop, and so the increased risk over appropriate time frames, such as 10 years, still have not been seen. Greatest associations I've seen in the first 5 years. And again, our finding that PPI use is a marker of cancer risk rather than a causative factor was one of the conclusions from the gut paper in 2006. Important to note, most PPI users do not show marked hypergastrinemia. That is, most of them have elevation of gastrin that are less than 2-fold. And while PPI use has increased, gastric cancers continue to decline. And so just lastly, I just want to leave you with some recent findings about the differences in effects of gastrin in the corpus versus the antrum. And so although the gastrin can stimulate proliferation in the corpus, and we've even had mouse models of hypertrophy and dysplasia, particularly in certain inbred strains of mice, most mice that have hypergastrinemia do not develop dysplasia. But interestingly, mice that are deficient in gastrin knockout mice almost routinely develop antral gastric tumors, including gastric cancer. And this was first described by Juanita Merchant's laboratory back in 2005, confirmed by us. We confirmed that mice that are deficient in gastrin, when treated with carcinogens, develop marked gastric cancers, as I'll show you. And thus, we've actually hypothesized that hypergastrinemia is protective against antral cancer. And what is the mechanism? The mechanism is that it turns out that the gastrin receptor is expressed in antral stem cells, which divide asymmetrically and lineage trace entire glands. And gastrin deficiency results in increased symmetric cell division. That is, instead of giving rise to a daughter cell and another stem cell, it gives rise to 2 stem cells. And this leads to increased proliferation and increased lineage tracing. And gastrin thus prevents cancer by inhibiting symmetric cell division and thus inhibiting a stem cell expansion. And we showed this very briefly in models of transgenic mice where they're lacking gastrin, so-called gastrin knockout mice, and giving back gastrin, infusing in gastrin via a gastrin pump, and showed very clearly that gastrin infusions reduce tumor number, they reduce tumor size, and interestingly, they also resulted in a 5-fold reduction in DNA mutations in the antral tumors, suggesting that there's a link between symmetric cell division, acquisition of mutations, and cancer susceptibility. So to just summarize in this part of the talk, gastrin stem cells are expressed near the base of the antral glands. They tend to be quiescent. They undergo asymmetric division. Carcinogenesis is associated with expansion of these stem cells, leading to proliferation, tumorigenesis. And interestingly, we found that carcinogenesis leads to a decrease in these niche gastrin cells, and that we think that antral atrophy and therefore loss of gastrin cells may be a key critical step in the development of antral cancers. So just to summarize what I've talked to you about in this talk, stem cells contribute to the late stages of ulcer healing. They're regulated by niche factors, including innate lymphoid cells. Hypergastrinemia secondary to PPIs can increase ECL cells through effects on corpus progenitors, causing their increased production and differentiation into ECL cells. PPIs are generally safe. They're overused, of course, but often given to patients with an increased risk of disease. And in mouse models, antral stem cells are suppressed by gastrin, such that hypergastrinemia likely inhibits antral gastrin cancer. So I'd like to stop here, thank members of my laboratory, former members of my laboratory, my funding and sources of support. Happy to take any questions. Thank you so much for your attention and for your patience. And again, it's a great opportunity to remember Mark Babyatsky, who was a dear friend, and we miss him very much. So happy to take questions.
Thanks, Dr. Wang. So please put your questions and comments in the chat, and Dr. Sands is going to help moderate with our questions and answer section.
Okay, I've been unmuted, so now I can speak in praise of a fantastic talk that really was very translational in nature. And Mark definitely would have appreciated your lecture and your very personal comments at the beginning. Please, if you have questions or comments, put them in through the chat and I'll convey them to Dr. Wang. While we're waiting for those questions for the next minute or so, I don't know if I can hold this up for you to see. It probably projects backwards the way cameras go. But this will be sent to you, Tim, to commemorate this. You as the 4th annual Mark Babyatsky Memorial Lecturer, and we're pleased to send this to you. So there are just a handful of questions coming in, and one is a comment from Sakshi Dua in our pulmonary division, who says that PPIs, at least the pulmonary folks, that PPI use is associated with lung non-tuberculous mycobacterium infection. So what do you make of the evidence of PPIs in association with lung infection in general, and maybe in particular with mycobacterial infection?
Yeah, no, sorry I didn't have time to go through all the PPI issues, but I think, as I mentioned, PPIs do result in hypochlorhydria. And there's been a fair amount of literature on pneumonia in general. The idea that, and it started, I think, with back in the 90s, and Bruce, you probably remember in ICU patients, the idea that patients in the ICU on PPIs, which were given for gastric cytoprotection because ICU patients are at increased risk for peptic ulcer and GI bleed. And so the idea that maybe it changes the microbiome of the stomach and even the esophagus, and therefore with aspiration, you're more likely to get a worse infection. So there's no question that acid has a physiological role. I mean, you can certainly block all acid and absorb your food, but I think going back, millennia, the idea was that acid is mainly there to sort of kill organisms. Bacteria actually is probably more worms and things than bacteria, but when people used to eat food that was not particularly clean. So it does have a purpose. And so certainly, theoretically, if you got or swallowed bacteria, particularly mycobacterium, those things could be regurgitated and aspirated. You can develop all sorts of different types of pneumonias. And so I think that literature has been mixed. Again, the problem, of course, is that it may be true, but it's hard to know from the point of view of the patients taking, so they're not randomized controlled studies. And I don't think there have been many that have looked at that in great detail. But I did have on my list this idea of atypical pneumonias, aspiration pneumonias. And the comment then about the atypical mycobacterium is quite interesting, and it's certainly a possibility. But I'd say that the data is not yet clear enough to say for certainty that that's a risk. And so, again, it's not to say that PPIs don't have any risks at all. But I think, again, it just points to the need that when we prescribe PPIs, that they're not completely innocuous. And I think that was the concern when these drugs were first introduced, that people thought that they had no possible side effects. But if you give them to 100 or 200 million people, things are going to pop up, and that certainly could be one of the risks.
Great. And maybe we have time just for one more question. And this is from Steve Itzkowitz, who, first of all, thanks you for a marvelous tribute to Mark and a great talk. And his question is Gastrin trophic to any other cells in the GI tract besides the ECL precursors?
So is it trophic? So I think, I guess the brief answer would be no, in the sense that it doesn't stimulate the proliferation of ECL cells or parietal cells. It does stimulate progenitor cells in the gastric isthmus. Now, in terms of being trophic, it does expand other cells besides ECL cells. these are not very well defined. One of them that's certainly believed to be true is parietal cells. Now, we haven't looked or studied or quantified in detail effects on these progenitors and whether or not they are the major parietal cell precursor or not. We haven't sorted that out. But I would say that it does lead to expansion of other gastric cells that we have not yet well defined. And those are studies that are ongoing where we're doing sort of more long-term studies on PPI treatment in gastrin on these gastric progenitors. And so hopefully we'll have more answers on that within a few years, at least in the mouse model. But I guess the answer is yes, not directly, but through the progenitors, yes.
Great. And I know we're just slightly over the hour, so I unfortunately, for the few other people who ask questions, I'm going to ask you to email Tim. There are only a couple more questions, Tim. So I really want to thank you so much for a spectacular lecture. Thanks for being with us to honor Mark. And hopefully we'll get to see you in person at some point in the coming year. Thank you so much. And thanks to everyone who tuned in this morning for the Babyatsky lecture.
Thank you. It's been my pleasure. Thank you, everyone.
Thanks, Tim.
Bye-bye now.
Thank you, Dr. Wang.
Thank you. Bye-bye.